Hello everybody, my name is Adrian Ryan. I'm here to discuss DNA fingerprinting and forensics. Firstly, I'll be talking about the outline of DNA and fingerprinting, and also the development and history of DNA and fingerprinting. After, we'll then take a look at how DNA and fingerprinting works on a molecule level and how it can be used. Finally, Laura will talk about the advantages of DNA and fingerprinting, the shortcomings of DNA and fingerprinting, and where DNA and fingerprinting could be in the future. Firstly, DNA stands for deoxyribonucleic acid. It is a hereditary material in humans and all other organisms. Everyone has DNA and it is located in a cell nucleus, or small amounts of DNA can be spotted in the mitochondria. When we talk about DNA, we know that it is contained in every human cell, and unique markers present within the genetic code allow for identification to occur. It is quite interesting that when looking for DNA, it can be found in quite a few places, such as blood, bone, mucus, tissues, skin cells, teeth, saliva, semen, and sweat, and human excretion. Most people will possess in each cell 23 pairs of chromosomes, half from paternal source and half from maternal sources. When examined at a genetic level, it will reveal a sequence of nucleotides that represents a DNA. Now, when looking at DNA and fingerprinting for forensics, we know that it relies heavily on the principle that no two individuals share the same genetic code. The test uses a DNA analysis and helps to resolve legal problems, identity in criminal cases when biological evidence is found at crime scenes, and identify victims of mass disasters and human remains. A DNA sample is measured by comparing the repetitive DNA located outside the coding region of the DNA. The repetitive unit is called polymorphic, as it can be different between people. DNA and fingerprinting is not only just used in forensics, it is also used in many other fields of work. It is used in paternity tests to help a single mother find out who the father is, or even to help a child to find their biological parents. They use it by comparing large numbers of DNA microsatellites to determine with high degree of certainty that paternity, maternity, or both. It is also increasingly being used in agriculture to verify certification, that is to know if a seed is really from the variety being claimed. Now DNA fingerprinting is being used on animals for parentage testing and to help identify and breed. The discovery of DNA and fingerprinting was founded by Alex Jeffreys in the 1980s on an unrelated project that he was working on, which was myoglobin genes in seals. When Jeffrey and his team compared myoglobin genes in seals to human counterparts, they found an interesting linkage. The short repeating sequences were similar in position and structure between seals and humans. When he compared those sequences with the published sequences of tandem repeats, called mini-satellites, it was seen to be the same result. They recognized that the repeating sequences could be highly variable informative genetic markers. With these results, Jeffreys and his group developed a radioactive probe made up of short sequences that could latch onto those repeating sequences. These tests revealed patterns that were unique to each individual, which was DNA fingerprint. He developed this technique with his colleagues after the Colin Pitchfork murder that took place. Pitchfork murdered for the second time in 1986. During the investigation of this case, it was discovered that he had the same identical method and blood typing matched the semen recovered from the scenes. The first application of DNA fingerprinting was method radio-labeled DNA probes, containing mini-satellites or oligonucleotide sequences. This method is based on restriction enzymes digestion, separated by agarose gel electrophorese, followed by southern blot analysis. The size of the DNA that is contained in restriction enzyme differ because of the variation in the number of repeated units. When excess probes are washed away, the exposure to X-ray films allow these variable fragments to be visualized and their profiles compared between individuals. In the early 1990s, DNA fingerprinting had evolved, and it was seen to be based on restriction enzyme fragment length polymorphism. This technique focuses on the repetitive sequences of DNA, which vary greatly from individuals to individuals. This method is also based on the help of using polymorph chain reactions because of the improved sensitivity, speed, and genotyping precisions. The DNA segments are separated by electrophores, which measure each of them by length. Once the segments has been sorted out by electrophores, a visual representation of the results are created by using a procedure known as audio-radio-graph. Now there is a more recent test that is used in today's society to measure DNA fingerprinting. One of the most recent tests is called short tandem repeat test. This test looks at the DNA segments and counts the number of repeats at a number of different sites. This current forensics test for DNA fingerprinting is based on a polymorph chain reaction. These chain reactions that is used in short tandem repeats is to create multiple copies of DNA sequences. The technique of using polymorph chain reaction in the short tandem repeat is to amplify the highly polymorphic DNA sequence with repeat units of 2 to 6 base pairs in length. The results are then separated and detected by using electrophores. Up to 30 short tandem repeats can be detected in a single capillary electrophore and will generate a unique code for each individual. Arthur will now speak about how DNA and fingerprinting works on a molecule level and how it can be used. Thank you.
Hello everyone, my name is Arthur and I am going to explain how DNA fingerprinting works in forensics and the current applications DNA fingerprinting can be used. When a DNA sample, for example a hair or a fingerprint, is found in a crime scene, it is immediately sent to the forensic lab where it undergoes various procedures. One of the common methods to identify fingerprinting is restriction fragment length polymorphism, or RFLP. The method relies mainly on the DNA base sequence that varies from individual to individual. This procedure utilizes specific enzymes to cut segments from a DNA sample in order to extract DNA. These segments are separated using a technique called electrophoresis, which measures each segment by length. Once sorted, another method, called autoradiography, produces an X-ray that displays an overlapping of the DNA samples and the results, thus allowing a conclusion of whether the suspect is innocent or guilty. Hello everyone, my DNA fingerprinting is used in a variety of ways in forensics. It can be used to identify an adopted or abandoned child's parents and their families through the child's DNA code sequence, which is a combination of both parents' DNA sequence. Another method is to diagnose an individual in order to find a cure for a genetically inherited disease. DNA fingerprinting is most commonly used to determine the identity of a deceased individual if he or she is present in a crime or a natural disaster, and helps recognize the culprit responsible for a rape or murder offense. Thank you.
Hello everyone, my name is Laura. I'm going to be discussing the benefits and advantages of DNA fingerprinting in forensics. The technique has been used since 1984, making it highly developed and improved throughout the years. For the subject being tested, it is easy and painless. It is said to be less invasive than taking a blood sample. It is also affordable and a reliable technique. It can be conducted in a relatively short amount of time. Anyone at any age can be tested with this method without any major concerns. And there is also a large variety of uses, such as legal claims, missing person cases, identification for the military, and paternity and prenatal testing. Hello. These are the main disadvantages and shortcomings of DNA fingerprinting in forensics. The sample of DNA can easily be ruined during the process of DNA fingerprinting, causing the sample to become completely useless for testing. The process itself is complex and tedious and can give results that may be hard to interpret. The test needs to be run on multiple samples, a numerous amount of times for ideal accuracy. Commonly, labs run each test twice with 4 samples. Privacy issues could occur if the information isn't kept secure at the lab. Personal information legally can only be released with a written order. This personal information, if leaked, could potentially complicate insurance processes, healthcare and job prospects for an individual. In this slide, I will be discussing the potential future for DNA fingerprinting. It is being debated whether or not the government should keep a DNA database of every citizen. The government would store everyone's DNA fingerprint in a computer software. Future tests will be able to show how a person will respond to different medications using simulations. We currently use chip technology to know which genes are active. This could, in the future, be useful to help create personalized treatments and cure the disease illness quicker. Could DNA in the future control our thoughts? It's a thought, isn't it? In this slide,